Hi guys, we are going to do genetic engineering now, okay? I hope you've prepared yourself. I hope you've watched all the videos on genetic tools because this is where it all kind of comes together, okay? So, uh, fasten your seatbelts. So, in the last video on the overview, we discussed this concept that in order to create a transgenic organism, you've got to take a gene from one organism out. You have to then package it up, put it in another organism, and when that other organism has a combination of its original DNA and new DNA, it is then a recombinant, or it has recombinant DNA, and it is a transgenic organism. So we're going to look in detail now at how those steps are carried out. Remember the reasons why we would want to make this transgenic organism is either to improve one of its features or to make it produce something that it didn't produce before in terms of a protein product. Okay? Right, so let's just break this down. We're going to look at this process in 3 steps. First is how to remove or isolate the gene of interest. So this organism has a gene that we want, and we want to put it in this organism. That's the organism that we want to improve in some way. So first we need to extract the gene from organism A. Second, we need to package it up into a vector. And third, we need to get it into the cell, into the organism that we want to, and and that is essentially producing our transgenic organism. So these are the 3 stages that we're going to look at, and hopefully, what I'm going to talk about, we can relate it back to these steps. Okay, so 1, extraction of the gene. 2, packaging that gene up into a vector. And 3, getting that vector into our second organism, thereby making the transgenic organism. Okay, so let's begin. Let's start with isolation. Okay, isolation of the gene of interest. Okay, I'm going to abbreviate that to that. Isolation of the gene of interest. This can be done in 2 ways. Okay, so let's look at method A. Okay, so in method A, so this is the one that's kind of described in terms of insulin, in terms of the production of insulin via a transgenic organism. But essentially, you'd have a cell, and you know that this cell produces lots of the stuff that you want, or it transcribes the gene that you want a lot. So it's very likely that this cell has a lot of the mRNA of the gene that you're interested in. Okay, so for our purposes, these could all be mRNA of insulin, of the insulin gene. So the mRNA is being transcribed. We would want to then find a cell of the pancreas that would be expressing this to high levels. No point looking for the insulin gene within the muscle cell because it's not going to be expressed in the muscle cell. Okay, it's in the DNA, yes, but there won't be any mRNA in the muscle cell because it's not expressed. Anyways, this cell has lots of mRNA. We can then isolate the mRNA, and use one of our genetic tools, reverse transcriptase. So reverse transcriptase will then copy the mRNA, okay, and then we could add DNA polymerase to then copy the DNA, the second strand back into DNA again. So So the first step would be to add reverse transcriptase. Once we've made one of the DNA strands, we can then add DNA polymerase to make a complementary DNA strand to the first DNA strand. So that then turns into this. Okay? So now we have our cDNA. We have a DNA copy of the insulin mRNA. Okay? So once we have that, that is not quite ready yet, because remember, if we want to put it into our vector DNA, we will need to add sticky ends so that DNA ligase can work. Okay? So what we can do then is add a few extra unpaired bases on this end, nucleotides on this end, and a few extra unpaired nucleotides on this end, thereby generating sticky ends that we can use later to insert into another piece of DNA. Okay? So this is essentially method one. You can do this another way as well. So this is the other way, and this is a kind of way that you can also use to essentially do the same thing. So what you could have again is you can have the DNA of a cell, okay? So you can extract the DNA, the whole genomic DNA of the cell. So that would be many molecules of double-stranded DNA, okay? And then you can treat with a restriction endonuclease. So it will chop all these big DNA molecules up into small fragments. It will still be double-stranded DNA, but very small fragments. But remember, because it's a restriction endonuclease, these fragments will already have sticky ends on them, yeah, because of the way restriction endonucleases work. But we don't know which of these is the fragment that contains our insulin gene. So what we can do is run all these samples we can put on a DNA electrophoretic gel. So we've treated with the restriction endonucleases. We're going to now run an electrophoresis and separate out the fragments by size. So we separated out all the fragments. And then what we can do is we can probe. We can add a probe. If we know what the sequence of our gene is, we can probe this electrophoretic gel with a labeled probe that has a complementary sequence to the insulin gene. So if it's giving off a green fluorescent signal, wherever that probe binds, to whichever of these bands it's binding, hope you can see this, to whichever of these bands it's binding and giving off that giving off that fluorescent signal or radioactive signal, we can detect that, and we'll know which of these fragments then contains our insulin gene. So we can just physically cut this out of the gel, extract the DNA, and we will have our double-stranded DNA with sticky ends, because remember we used a restriction endonuclease. So we just cut out the DNA and it will already have sticky ends. And we know that this DNA is for our insulin gene because we have probed our electrophoretic gel. We've probed all our fragments with a probe specific to the insulin gene. So these are the two ways you can end up isolating your gene with sticky ends ready to be inserted into a vector. On we go to the next stage. So we've done stage one, we go to stage two now. How do we package this up into a vector? So we're talking about stage two now. Stage two is stabilizing our DNA, okay, because these short fragments of double-stranded DNA with overhangs, i.e. sticky ends, they are not very stable, so we need to stabilize it and package it, stabilizing and packaging into a vector, okay? And in most cases, a vector will be a circular piece of DNA known as a plasmid, okay? But other vectors are possible, and I suggest that you look into this. But I think for our purposes right now, we want to give context to this idea of getting DNA from one organism into another. Once we've understood that in the most basic situation, then you can go on to look at the more, or the modifications or the different circumstances for yourself. Okay, so we've got our gene of interest, and it's got sticky ends, okay, and now it needs to go into a vector. Now, what is a vector? A vector is a circular piece of double-stranded DNA, okay? Now, usually plasmids are already present in bacteria, okay? So you can get naturally occurring plasmids, and it's a way for bacteria to kind of exchange DNA with each other. Usually these plasmids contain genes for antibiotic resistance on them. So if one bacteria is resistant, they can undergo bacterial conjugation. The plasmid, the genetic information on the plasmid can be passed from one cell to another, and so resistance can spread from one bacterial cell to another, independent of division. Okay, but you can also synthesize these plasmids. Scientists can synthesize them themselves by joining various pieces of DNA, so they can be designed for this purpose of genetic engineering. Okay, so the basic plasmid will be a double-stranded piece of DNA, and the most important feature for us is that as part of it, it will have genes for antibiotic resistance. So this gene right here will give whatever bacteria has this plasmid, that bacteria will have the ability to be resistant to, for example, ampicillin. Okay, this is an antibiotic. Okay, and it will need a second antibiotic resistance gene. So this gene might give the bacteria resistance to tetracycline. So ampicillin resistance, and this gene will give it resistance to tetracycline. Tetracycline resistance. Now, the way that we get this gene of interest, so let me just label this, the way we get this gene of interest into the vector is using, again, one of our tools, restriction endonuclease. And if we're smart, we'll use the same restriction endonuclease that we use to isolate this gene, or if we use the first method that we talked about, the sticky end that we've added on here will be the same that is produced by the restriction enzyme that we're going to use now. Okay? So what we'll do is we can treat this vector with a restriction endonuclease. So we need one of our tools, restriction endonuclease. And because we're smart, we will use the restriction endonuclease that will produce just the right sticky ends that could anneal to our gene of interest. So remember, in our example, what we've been using so far is echo R1, and that produces the sticky end. That produces the sticky end TTAA. And on this side again, TTAA. So if we cut this plasmid with a restriction endonuclease, get rid of these for now. Okay, if we cut this plasmid with restriction endonuclease, we are going to generate sticky ends. Okay. TTAA here, and here TTAA. Yes, no. TTAA here. Okay. Now, hope you can see that if we take this, if we mix these two fragments now, what will happen is that this sticky end will anneal here, and this sticky end will anneal there. TTAA, TTAA, AATT, AATT. Thank God for high def, right guys? Okay, so if we stick this here, maybe I should have used different colors. Anyway, if we stick this here now, one of the sticky ends will anneal here, the other of the sticky ends will anneal here, and we have inserted the gene. It's now annealed. So restriction endonuclease, we treat the vector with the restriction endonucleases. We mix the DNA from different sources. Okay, so essentially for us that's our gene of interest plus our vector plasmid. Okay, and what happens is the gene of interest, so the sticky ends allow annealing, so just rephrase that. Because we've used the same enzyme to treat both fragments of DNA, we have sticky ends that allow annealing of the DNA, okay? And then in order to finally reform a continuous DNA molecule, we add another of our important genetic tools. We add DNA ligase, because remember it's only hydrogen bonds keeping the one fragment attached to the other fragment. We need to reform a continuous sugar phosphate backbone. So in order to do that, we need to treat with DNA ligase, and when we do, these will join up, okay, to form two continuous DNA strands linked together by hydrogen bonding. Perfect. Okay, so there we have it. We've now completed stage 2. We've stabilized and packaged our DNA, our gene of interest, into our vector, and we should note at this stage that we have just produced our recombinant DNA. Okay, we've taken DNA from one source and and added it into our plasmid. The DNA are from different sources, joined together, recombinant DNA. But we haven't yet produced our transgenic organism because it's not yet in the cell that we want it to be in to be expressing the gene product. Okay, so that's stage 2 completed. We'll now look at how we can get this DNA into our recipient cell so that it can be expressed and we can go home. Okay, we are back. So now, once we've made our plasmid, and we've got our gene of interest, we have got it nice and packaged up in our vector. Now it's time to get it into our recipient cell, and in the simplest case, that recipient cell is a bacterial cell. Okay, so we can have a few bacterial cells for this stage. Okay, and just shrink that down a little bit. Plasmids are much smaller. Okay, so the next stage is to get our recombinant plasmid into a bacterial cell so that the bacteria can start expressing the gene product. However, the first thing we have to appreciate is that not all of our plasmids are going to be recombinant. Some of our plasmids are just going to be regular, they're just sealed up back onto themselves. Okay, so the way that these DNA is taken up by bacterial cells is essentially because bacteria naturally have an ability to take up extracellular source of DNA as well as via conjugation. And this is not exactly the same as conjugation, but I think that the property of taking up DNA from external sources is linked together with that. Okay, so this is, I mean, I guess through evolution it has been an advantage to bacteria to be able to take up DNA because it gives it the ability of expressing genes that it didn't have before, and in some cases that might have led to advantage for the bacteria, an evolutionary advantage. Okay, so anyway, let's begin. Now, in the presence of calcium ions and something called a heat shock. So that's basically, it's going from very low temperatures to, you know, much higher temperatures very quickly. Under those conditions, bacteria tend to take up DNA a bit more. So what you can have is you can have cells that then take up some of these bacterial, some of the plasmid vectors from the extracellular environment. Okay. Okay, so this can happen. All right, so then we remove the bacteria from the heat shock environment, and basically then we just have to plate the bacteria on agar, which contains the nutrients for the bacteria to grow. Why do we use bacteria? They have a really short generation time, so within 20 minutes you have one generation becoming 2, and within a further 20 minutes those 2 cells have become 4, and so on. So within a matter of hours you can have thousands, millions of cells. But the complicating factor is that not all the cells have taken up the plasmid, the recombinant plasmid. Some cells haven't taken up any plasmid. Some cells have taken up plasmid, but they haven't taken up recombinant plasmid. So only a small fraction of all the cells that we use will be transformed, transgenic bacteria. Okay, so let's just say that when bacteria take up external DNA, they are said to be transformed. However, it's only those cells that have taken up the plasmid with our gene of interest that can be described as transgenic. Okay? Anyway, how do we find the cells which are transgenic and not any of the other ones? Okay? So we need to keep this idea that some cells have taken up the plasmid that is transgenic and others have not. How do we differentiate between them? Okay? And what we need to remind ourselves is that our plasmid had 2 genes for antibiotic resistance. One of the genes gave it resistance to ampicillin resistance gene, and the other gene had resistance for tetracycline. It's called TET. Resistance. Okay? Now, this is the clever bit, because the restriction site that the fragment gets inserted into is in the middle of one of the genes for antibiotic resistance. So when, in the case that we have a plasmid that does have the, or when we have a recombinant plasmid, the situation looks like this. So what we have is our gene of interest inserted right bang in the middle of our gene for tetracycline resistance, which kind of messes up our tetracycline resistance. However, the vector does give resistance to, still give resistance to ampicillin. So our recombinant plasmid is only resistant to ampicillin, whereas a plasmid which is non-recombinant is resistant to both ampicillin and tetracycline. I hope you can see that. So what we then do is we then plate these bacteria on an agar plate. Okay. So the next thing that we do is plate the bacteria on agar. Agar is a jelly-like substance, or it's jelly-like when it's cool. But what it does have is nutrients that promote the growth of the bacteria, allows the growth of the bacteria. Okay, so if we plated all the bacteria on the agar, it would look like this. Okay, we get lots of colonies everywhere. Okay, lots of colonies, each colony being derived from a single bacterial cell that over 24 hours has gone from 1 to 2 cells because of binary fission, 2 to 4, 4 to 8, 8 to 16, 16 to 32, and so on, until after 24 hours, I think we're talking about possibly millions and billions of cells, okay, in that one colony. But what that means is we can see it by eye. We can see that colony by eye. Anyway, that's on regular agar. Okay, right? However, if we had plated those same bacterial cells on agar that contained ampicillin, an antibiotic, the only colonies that would grow would be the bacterial cells that had been transformed with our vectors. Okay? But of all these colonies, we don't really know which one of those contains the recombinant plasmid and which one of those contains the regular non-recombinant plasmid, until we lift these colonies exactly in the same position by pressing something down on this plate and then pressing down on this plate, so we transfer the cells from there to there, and this plate contains agar plus tetracycline. Okay, now this time we'll get only a few colonies here, but there'll be one colony missing. Where did this colony go? Right? That's the question. Why did this colony grow here but not there? The only conclusion can be because the cells here were resistant to ampicillin but not to tetracycline, i.e. it must have been because they have taken up the recombinant plasmid only. Okay, which means that they were resistant to the ampicillin, but when they were transferred to the tetracycline plate, they were not resistant to that because the tetracycline gene, tetracycline resistance gene here has been disrupted, and recombinant plasmids don't confer resistance to tetracycline. So we have therefore identified that this colony is a transformed, transgenic colony of bacterial cells. Okay? And there we have it. So once we've isolated that, we can lift some cells from here, we can grow them up in larger volumes, and just very briefly, you already know what's going on, but inside these cells, if we look at just one of the cells, what is going on, and this is important to understand, that because that plasmid is there, because it has our gene of interest, that gene of interest in the bacterial cell is being converted or copied into mRNA thanks to the RNA polymerase of the bacterial cell, of the recipient cell. RNA polymerase is transcribing the gene. The ribosomes are then translating the mRNA to make protein or whatever product, and that protein then goes on to either give the cell a new modified function, either some new modified function or characteristic, or it provides some kind of product that we can isolate and use. And ladies and gents, that has been genetic engineering. If you have any questions, do put them in the comments below, but I suggest that you really try and figure this out a little bit for yourself. It's the best way to learn these things. But yeah, if you've tried your best and you still have questions, I'm happy to answer them. Good luck, guys.